So to begin, this is an image of brain vasculature in a mouse's brain. As we scroll through, you're actually moving deeper into the brain, and that's how we see the change in depth, and we can also see a change in time. So the green outline is specific to a vessel that we're looking at this point in time. As you can see right here, this long piece here is white, which indicates the fluid in your blood, and this little black spot here is actually a red blood cell that is stalled. So that means that it's not flowing, and we can concur that the entire vessel is not flowing. So in this case, since we see that this white is interrupted and there's this black spot here, we can consider this entire vessel to be stalled, and therefore we would label it as a stall and later mark this spot as the point of stalling. Here is another beginner video. As we scroll through, we see the vessel outline show this piece of white, and again you see the black here and a secondary black area here. So in the game you would mark those two spots as stalled, but either one is an appropriate choice. And another beginner video. As we scroll through, we again see the outline around this white piece of vasculature, and again we see the black spot here. I believe there was another one down here. As you can see, those are stationary throughout moving through the video. To contrast, if you look at the vessel up here, we can see the little black spots moving as I move the scroll bar. But when I stop the scroll bar, you see black spots such as here and here. But that does not mean that it's a stalled vessel. You have to look at the video as a continuous flow, and that will be your indicator as to whether or not a vessel is stalled. Example of a stalled beginner vessel. As we scroll through, we can see the outline vessel that the green outline is trying to indicate. And as we stop here, we can see these black spots here, which represent the red blood cells that are not flowing through the vessel. As we scroll up and down, those spots do not move. So that indicates that the vessel is stalled. Another stalled beginner video. As we scroll through, we see the vessel that the green outline indicates. This curving structure is fairly normal, even though in other images we regularly see fairly linear vessels. As mentioned before, the black dots indicate stalling, and because they stay in the same points throughout the video, the vessel is considered stalled. It's a beginner stall. As we see here, the green outline indicates this vessel piece here, and there's the black spot representing the red blood cells stuck right here. And I'll go up and down to demonstrate the fact that it's not moving. Another stalled beginner vessel. As we scroll up, we see the vessel indicated and the black spot not moving. To remind you of what defines a vessel, it's a point that has two other branching points on either end. To demonstrate, on here we can see that there's a branching point coming off here and here, which indicates the end of this piece of vasculature. And up here we see it again branch into two directions. And that's how we define this outline here. Here's another example of a beginner video. As we scroll through the vessel, we again see the black spots not moving in the white flow. An interesting thing about this image is that the contrast is a little off, so the entire image is fairly bright. Again, this is very normal, but this is still considered a beginner image because these pieces of black in the middle are stagnant throughout the entire image. Another interesting thing about this image is this line up here. This is again due to stitching of the images that occurs during our processing, and it's very normal to see, and it shouldn't cause any problems with the image analysis process. This is another beginner video. As we scroll through, we see several instances of the red blood cells being stuck in the vasculature, as shown here. One thing that might be a little confusing to users is this line, which previously mentioned just indicates where the stitching of the images occurred. And it's normal to see and shouldn't interfere with any of your analysis. Another problem you might run into is this little piece of vessel here. It is not in fact a stall up here. Instead, it's a piece of vasculature that bends and comes back up. If you can follow this piece here, if you watch it as I go back down, you'll see that this whole piece of white is continuous through to that little piece that you see right here. This is another beginner stalled video. But the big question in this image is, which vessel is this green outline indicating? As you can see here, it looks like maybe this piece of vessel is what it's trying to get you to look at. But in reality, it's this image right here. So this vessel right here, as I flow up and down, you can see it travel throughout the piece of green. And the way we know that is because if you look at the original one I showed you, If I look at the white here and then I follow where it goes, you can see it up here, traveling further, and I'm going to keep going. You see it go up here. At this point, it should be branching off into two different directions. Instead, it keeps flowing up over here, which shows that the branching point did not occur at the end of the green outline. And that's how we know that that vessel wasn't a piece of vasculature that we wanted you to analyze at this point. So instead, as you look at this image here, the piece of vessel we want you to see, you see the branching point here into two different directions. And again at the bottom, you see it branch in two different directions. This is another beginner stall video. As we see here, we see the piece of white with a black dot in it that isn't moving as we scroll through. Something to take note of here is this large vessel here. That's something that you'll see every once in a while in your videos. And in case you're wondering, we don't generally assess these vessels because they're so large that they're almost never stalled. And this is because these vessels over here that we actually look at are about the width of a red blood cell, and that's why they can stall more easily. But in this instance with this large vessel, the diameter is so much larger than that of a red blood cell that they never get clogged. Another beginner stall video. As you can see as you look through the image, there's the black spots that don't move. An interesting thing about this image is that it has a somewhat U shape, which results in the green outline being more circular as opposed to linear. And that is normal to see as well, especially when you see a curving structure of the vessel.